I would first of all, Ildiko, like to introduce you to Father Greg Bazilla, who is the priest at Holy Trinity Episcopal Church in South River, and who I have the privilege of serving with. And same goes for you, Greg. This is Ildiko. I've known Ildiko now, she's going to probably correct me, 12 years, is it?
At least.
At least, if not more. But I want to welcome you, and again, to reiterate what Father Greg said, to give thanks for your gift of time and talents on this subject. So I'm just going to give a brief intro, Ildiko. Dr. Ildiko Antal, head of non-clinical project management at J&J Pharmaceuticals. Dr. Antal, who has 30 years experience in drug development, will make a presentation on the COVID-19 virus, the mode of the action of the COVID-19 vaccines, the different types of vaccines and treatments that are being developed, and why everyone should be vaccinated. There will be a time for questions that she will try to answer the best she can. Thank you so much, Dr. Ildiko.
Thank you so much for the kind introduction. I am really happy to be here. I am actually a neuropharmacologist by training. I am not a virologist, but after many years of working at the bench, I became actually a project manager. And as a project manager, managed a number of different projects through development and launch. And most recently, I am working for Janssen & Johnson, and I am the head of non-clinical project management there. And my team also participated in the development of the COVID-19 vaccine, which is currently authorized for emergency use, both in the United States, in Canada, in the European Union, and also in Bahrain. So far, but it will be filed pretty much in almost every country in the world. So let me share my slides with you. I am, can you just confirm that you can see it? And I am going to show you 12 slides, and after this, there will be certainly enough times for questions, which indeed I will try to answer the best I can. So the status of the COVID-19 COVID-19 virus infection as of yesterday, that in the world, there are almost 120 million cases. There are more than 2.5 million people who unfortunately perished in this terrible disease and pandemic, and about 96 million of them recovered so far. Of course, these numbers are never accurate because different countries will have a different ways of reporting, but it certainly gives us a very stark view of what's going on right now. And I said to say that after reading the account by John Barry about the great influenza between 1918 and 1920, I have to say that unfortunately, especially the United States government and many of the people who are taking care of us with respect to our health have not really learned anything from this historical event 100 years ago. And unfortunately, we repeated almost all exactly the same mistakes that actually have happened in the 1918-1920 influenza. The only big difference is that the number of people perished in this pandemic is actually much fewer. And the reason being is because the state of the healthcare is obviously different than 100 years ago. But nonetheless, it's 2,650,051 people more than it should have happened. So just a general account about the sequence of events and a little bit about the virus itself. So the COVID-19 virus sequence was published on January 10, 2020, and the antigen started to be developed within days. After this, there was a very, very collaborative effort between academic organizations, industry, government, especially the United States government, but also some other major countries' government, and the World Health Organization to actually start to develop vaccines against this virus. There are more than 90 different vaccines are being developed in the world. And they are having a number of different kinds of mode of actions, which I will talk about a little bit in subsequent slides. There are 23 companies that are working on COVID-19 treatments. So these are treatments that could help the people recover faster who are already ill. About the coronavirus itself. So the coronavirus is actually a class of viruses. And what you need to know about a virus, that a virus is basically in the liminal space between living and non-living things. A virus is actually a very simple construct. It has either a piece of RNA or a piece of DNA in it. So there are basically two major classes of viruses, either RNA or DNA based. And besides that piece of genetic information, all it has is a bunch of protein which is protecting that piece of RNA or DNA. And the RNA and DNA in the virus basically just codes the protein that is protecting it. That's all. And what it does, that it gets into an organism, which could be an animal or even a plant actually, or human beings. And then it basically just replicates itself. And it uses all the biological material of the host organism to being able to replicate itself and recreate its own RNA, DNA, and its own protein. That's all it does. And so it's a really wicked thing. And so the coronavirus is one class of viruses. It's an RNA-based virus. And like the SARS-CoV-2, which is actually the COVID-19 virus, or the SARS virus or the MERS virus, which also attacked the civilization a few years back, they are all coronavirus types. And there are other kinds of viruses, like rhinoviruses, which cause the common cold, or there are adenoviruses, etc. The COVID-19 virus is actually more contagious than the SARS and the MERS virus. So when the SARS and the MERS hit, pharmaceutical companies also started to develop vaccines against them, but they didn't become a pandemic. So the reason for that is because it wasn't as contagious, and wherever it hit first, the countries were able to contain them, so it didn't become a pandemic. You also need to be aware, especially in the early times, maybe not so much today, that there were many different antibody tests were available, and many of them were not completely fully validated, and many of them, unfortunately, still today create either false positive or false negative results. So you have to be actually very careful, making sure that you are using tests which are like official or fully validated in order to be able to really believe the results. As you know from the news, there are also a few mutations already created for this COVID-19 virus. Viruses are mutating easily. The reason why it happens is because, as I said, they replicate their own piece of genetic information in our body. And unfortunately, when they do that so many times, they actually make a mistake. Mistakes just happen. It normally happens also in biological systems. It's not completely mistake proof. And so when those mistakes happen, then a mutation will emerge. And some of these mutations can be more contagious, for instance, than the original virus was. And that is why it is so important that we actually have to reach herd immunity against this virus as quickly as possible, because this is the only way that we can slow down that it keeps mutating in the human body and becomes even more viral or more dangerous than it was originally. And in the early research times, when we created the first vaccines and started to use them in animal studies, We also had to study, what is the physiology of the virus and what really is happening with that. And so we had tests in non-human primates, which can be infected with COVID-19 and develop very similar symptoms as humans. And in the early studies, we could show that if they recovered from the illness and they were reinfected, they didn't get sick again, which was actually very, very encouraging results at the early times, which basically helped us to understand that if we develop a vaccine, then it probably indeed will be efficacious. So I want to debunk basically the first concern that the general public had about the speed of the vaccine development. So people are very concerned about, that these vaccines may not be safe because they have been developed so quickly. So the honest truth is indeed that when you are developing drugs, which this scheme shows the steps of drug development in general. It starts with research, then there are the early development phases, which are the phase 1, phase 2 clinical trials until you prove the concept. And then once the concept is proven, then you move into large phase 3 clinical trials or more, and then you submit it to the regulatory agencies or health authorities, and finally the drug is going to be launched. Now, the early development usually in a small molecule, and vaccines are large molecules, they are a little bit faster. But for small molecules, it could be over 6 years, and it will cost about $650 million. The clinical development part can cost another 7 years, which can be up to more than $1 billion. And then the launch itself is probably going to take another year sometimes, and it will cost another $500,000. So this is a typical scheme for usually a small molecule. For vaccines or large molecules, it's normally a little bit longer, but still the early time can take about 1 to 10 years, and then the later stages a couple more years, and again the launch. Now, in case of the COVID-19 vaccine, we actually managed to get from the cloning, which I told you that it was in January 2020, until we actually were able to start phase 1 trials in 8 months. And this is why we were able to do that. Normally, when you are running a drug development process, usually, there is an unmet need, but there isn't a huge rush. So you know that you have some time to actually develop maybe another drug for a disease which may be already treated by something else on the marketplace. And then usually what you do is that you take all the different studies and steps, what you have to do, and you do them sequentially. So you make the first one, wait the results, you evaluate the results, you take the next one, et cetera, et cetera. But in this case, we didn't have that much time. So basically what we had done is, first of all, we put a lot of people on that, not just our company, but every company, put a lot of people on it, put a lot of money on it. We did most of the things that normally done sequentially at the same time. And the biggest time saver is actually manufacturing. So in a normal situation, you would do all the different studies, wait until all the results are labbed up, and then you're completely, absolutely sure that this is the compound, or this is the vaccine that you actually want to file in the regulatory agencies and put on the marketplace. And then you start manufacturing it. And manufacturing is also taking quite a long time, right? You have to do a lot of tests and this and that and the other, and it takes a long time to actually produce the required amount of medication. In this case, we actually developed 3 potential constructs that could be a vaccine at the end. The reason why we were so familiar with that, because we used the same constructs that have been used for other vaccines in my company. So we used the same construct that has been used for Zika, it's being used for HIV, and it's being used for the Ebola virus, which is already approved, that's on the marketplace. And this construct has been studied with hundreds of thousands of patients for many years. And what actually could give you any side effects or issues or safety signals is the construct itself, not the actual DNA what we are putting in there. So we were very confident that the construct that we are using is already true and tried and safe. And once we had the three different constructs that we thought could work at the end and will be efficacious, we immediately started to manufacturing them. So we manufactured them at complete risk. And as the studies progressed, and it was clear that we just have to use the one that is going to be the winner, then obviously we just abandoned the other two and ramped up all the manufacturing of the one that is actually ending up on the marketplace. So that is a huge amount of time savings, and it's also a huge amount of investment actually at risk. And we really had to do that for humankind in this moment of time to make sure that we are going to rush the vaccine to the marketplace as quickly as possible and make sure that it is as safe as possible. And again, it was done by a very, very strong and large collaboration with the Food and Drug Administration in the United States, with BARDA, which is the Biomedical Advanced Research and Development Authority, which is a government agency in the United States. And of course, as I said before, we also work together with the World Health Organization. We did all the non-clinical, clinical testing and the manufacturing parallel, as I said. The virus vector was very well known because it has been used in all these different other vaccines that we have been developing and already put on the market. The scaling and the manufacturing was all done at risk. We also did a lot of collaboration with other companies to manufacture the vaccine. And you might have heard that most recently, which is a really unheard of and unprecedented and pretty amazing collaboration between two competing large pharmaceutical companies, that Merck, who actually also developed a vaccine, but unfortunately their construct wasn't efficacious. So they actually had to abandon it from the clinical trials. They offered that they take our proprietary vaccine and they are manufacturing it themselves. So actually Merck is manufacturing the Johnson & Johnson vaccine, and that's why it is going to be available also like about 100 million doses probably by June, and hopefully 1 billion doses by the end of the year. And we have to file these drugs, all these vaccines for emergency use application, which means that it is actually being filed before the length of the trials normally complete. So normally these vaccine trials would run at least 2 years, sometimes longer, it depends on the virus. And in this case, these trials were actually stopped after were tested, in our case, in about 43,000 patients, because the incidence number was high enough that they were able to make a confident statistical analysis in order to determine that actually the vaccine is efficacious. And then you can file the emergency use application at the Food and Drug Administration. I also would like to let you know that the way how the drugs are being evaluated at the FDA is extremely rigorous. When we actually file the emergency use application or any application for any drug, you actually have to provide all the raw data, every single data point to the Food and Drug Administration. And what they do is that they use their own statisticians, so the Food and Drug Administration statisticians, and they completely independently, using your raw data, they reanalyze everything that you have done to make sure that the statistical analysis is actually correct. And if it matches with the company's data analysis, then they will assemble an advisory board, which is usually 15 or 13 or 15 members of subject matter experts that can evaluate all the data and the results. They will listen to the company's data analysis, and they also listen to the FDA's data analysis, and then they will make a recommendation whether they recommend the drug for approval or not. And the FDA is not bound by the recommendation of the advisory board, but usually like 95% of the time follows it. So in this case, The Pfizer, the Moderna, and the Johnson & Johnson vaccine were all applied for emergency use application at the FDA. And there were advisory boards in all 3 cases. And in all 3 cases, they actually recommended for approval, and they were approved for this use. So how does this virus actually causes the disease? So the way how it actually gets into the body is through, it gets into the lung through the mouth, nose, eyes, and from droplets in the air. The way how it gets into our cells is actually using a receptor within our cells. It's true for every virus, and it basically is an evolutionary thing, that the way how viruses can survive in other living beings is that they found a way in, and they usually latch into something which is already in those living beings. So in this case, they actually cause the adenine converting enzyme 2 receptor, or ACE2 receptor, in order to enter into the body. And the lung has this receptor in very high numbers. You also must have heard that some people who have underlying health conditions are more susceptible, or elderly, more susceptible for the virus. And the reason is because the people who have high blood pressure and being treated by ACE inhibitors, they have actually higher number of ACE receptors in their lungs. Also, as we age, the number of ACE receptors just increase in the body. That's just the natural way of our biology. So that's why children are less susceptible to the virus and less being infected than elderly people. Now, once the virus gets into the cells, it releases its piece of RNA, as I said, and it uses the cell's machinery to synthesize its own protein and assembles into a new virus, and the whole cycle starts again, and it's just keep replicating. But the body is not going to just watch this happening. We have a very strong and complicated immune system with a lot of different immune cells, and we are actually creating a response against this intruder, and we are going to naturally develop antibodies against it. The problem is that, you know, if the virus is coming at us in really large numbers and our immune system cannot keep up with that, then it basically overworks itself. It goes into an overload, which is actually ending up in inflammation. And that is the inflammation which is creating the problems in our lungs. So when that inflammation happens, then actually these are all the alveoli at the end of the trachea and all the different tubes within the lungs. The high level of inflammation is actually going to cause a lot of fluid increase. And that fluid is the one which is actually causes the death ultimately, because the patient will not be able to breathe. And unfortunately, this is the same mechanism. So almost all respiratory illnesses or other kind of virus illnesses, including the influenza itself. So what is herd immunity, which you also have heard in the news probably many times? So it is when 70% of the population will be protected from the infection. And the way how it can be protected is either by developing antibodies against the pathogen, or getting antibodies like President Trump did when he got sick, then he got actually antibody treatment, or the people who can get vaccinated and again develop their own antibody that way. So there are a number of different types of vaccines that can be developed, and this actually shows what kind of vaccines have been developed for the COVID-19 vaccine. The most ancient type of vaccines that in the early research time, you know, like even in the early 19th century started, was from inactivated virus or inactivated bacteria in some case. And for instance, the Chinese COVID-19 vaccine, which is already available in China and several countries in Europe, that is an inactivated virus. The other kind of vaccine could be the weakened virus, which in this case, you know, like it was developed for MMR and smallpox and chickenpox, which we already got when we were children. The next kind of virus is the vector-based viruses. These are viruses that have, vector-based vaccines. These vaccines actually have a construct in it. The construct starts with a non-replicating virus. Oftentimes it's an adenovirus, which we used and other companies as well. And this construct will have the piece of DNA of the protein, which is the targeting protein of the virus. So in the case of the COVID-19, that's the spike protein. The spike protein is the one which is going to latch on that ACE2 receptor in our lung, and that's how it's going to get into the cell. So if we just isolate that one piece of DNA that codes that spike protein, and we put that into a non-replicating adenovirus and use that to get into the body, then once it gets into the body as a vaccine, then it releases the DNA from which codes the spike protein. The DNA is going to be translated into an RNA, and the RNA is translated into a protein, which is the spike protein. It is the same protein that the virus itself has. And the body naturally is going to develop antibodies against that protein. And this is the way how the body is going to be protected against the regular COVID-19 virus infection. And the last virus type is the RNA-based viruses, or the DNA, depending on what the virus is. RNA and DNA-based vaccines. And the Moderna and the Pfizer vaccine are RNA-based vaccines. They actually just a piece of RNA in some protective material, they inject it into the body. The RNA is going to be translated into the spike protein, and again, the body is going to develop antibodies against it naturally. So the difference between the Janssen vaccine and the Pfizer and the Moderna vaccine is that the Janssen vaccine is in a virus construct. It is a lot more stable. It is probably give you longer term protection. And because it's much more stable, it can be put only in a refrigerator, and it's stable in a refrigerator for like 2 to 3 months, in a regular freezer for several years. And so it is much easier to distribute. The Pfizer and the Moderna RNA-based vaccine is actually a little bit of more unstable construct. And that's why it has to be kept in a -80 degrees centigrade on dry ice and in liquid nitrogen or whatever people have. And so that actually makes it much more difficult to distribute. And also because of it's not as strong or stable, then we actually need 2 doses from that to reach full protection, while the Janssen vaccine is able to protect you actually after 1 dose. And the original data from the Janssen vaccine showed that it was, I believe, they said 72% protective. It's actually, as the trials go on, and they are continuing up to 2 years, the data is actually becoming stronger and stronger because the incidence number goes up and the statistical analysis is better and better. So actually, the Janssen data is really giving you very, very strong response as well. And it is coming from just 1 dose, which certainly has an advantage as well. So I want to show you a very short video, which is going to show you what I just said, that how the vaccine works in the body. Actually, you can't hear it, right? I have to make sure that I turn on the computer sound too. Okay.
Vaccine, which uses an adenovirus as a vector. Adenovirus causes the common cold, but the vector is genetically changed in the vaccine, so it cannot replicate. Once the adenovector vaccine is injected into the body, the immune system starts making antibodies. These specific antibodies work to attack the piece of the coronavirus. Then, once the perceived threat is gone, the immune system creates memory cells. Memory cells remember how to fight the virus. So if and when they encounter it again, the immune system is ready to fight it off. This means the body is immune. Once
Okay. And so now I just wanted to address a couple of other questions that people usually ask. Very often I hear, does any of the vaccines will alter my DNA? It is an absolute resounding not. The vaccines are made to use your cellular material to create their own spike protein in your body, so you naturally can develop antibodies against them. It is not building into anybody's DNA or anybody's genetic material. It has nothing to do with that. Doesn't matter who says that, whether you read it in any social media sites, it's actually scientifically untrue. Why should I be vaccinated? Well, in order to protect yourself from severe disease or death and reduce the chance that the virus can mutate and become more deadly, that is really the main reason why we all should be vaccinated. It is actually really important that not just in the United States, but the whole world should reach herd immunity, so we will, conquer this virus, and it's not going to continue to be deadly all over the world. Many people ask that if you have a vaccine which has 2 shots, like the Moderna and the Pfizer vaccine, why is that you have symptoms after the second shot? The reason is that after the first shot, you already reach some level of immunity, but they determined in their clinical trial that immunity wasn't complete or wasn't satisfactory. So that's why they suggested to get a booster shot. This is not uncommon. If you have gotten other vaccinations like tetanus, for instance, or the shingle vaccine, or many others, you actually do need some booster shot throughout your lifetime of those. But your immune system is already working hard, and when you get the second shot, then it actually goes a little bit of an overwork, and that is the reason why people usually develop fever or maybe nausea and these kind of things, but it usually doesn't last longer than for a day or a day and a half. And people also ask that why do they need to wear masks and do social distancing after they are vaccinated? Well, the reason for that is because you still can get infected. So just because you are vaccinated, if you meet somebody who actually carries the virus, you still can get infected. And then once you get infected, you are not going to get sick because your body is already protected and it will protect you against the virus, but you still can shed the virus in your droplets. So when you cough or you sneeze or even just breathe, and then you are close in close proximity with somebody else, and if that person is not vaccinated, then you will infect that person and that person is going to get sick. So that's why it is very important that we all have to vaccine until we actually reach at least herd immunity. And this was my last slide. So I hope that it was helpful for you. And then if you still have any questions, I am happy to try to answer them.
There are a couple questions in the chat window, I see.
Okay. Let's see if I can find that. I have to stop sharing, I think. Yeah. Okay. Okay, we live in New Jersey, headquarters New Brunswick. What kind of work on the vaccine is done in us? Also in New Jersey, manufacture vaccines there. So the Johnson & Johnson vaccine development, Johnson & Johnson vaccines development is actually in Leiden, Holland. So all the studies and all the non-clinical and discovery studies were done in actually Leiden. The clinical trials have been done all over the world. So it was in certain countries in Europe, in the United States, in some countries in South America, and also in South Africa. So the Johnson & Johnson vaccine was also tested against a number of different strains of the virus, and it was found to be effective as well, which is also very encouraging. And there was one more thought I wanted to say on that. That some people were concerned, that the effectiveness of the overall effectiveness of the Janssen vaccine is 85%, and the Moderna and the Pfizer vaccine is 95%. So that must be much better. You don't know that. They were tried in very different circumstances, in very different cohort. And the only way you can tell whether one vaccine is better than the other if you actually test them against each other in the same trial. So those numbers by themselves really don't mean anything. It's like compared really apples to oranges. They all effective and they all safe, and that's great. But you cannot say that one is better than the other, to be honest with you. And with respect to Merck, so Merck is indeed in New Jersey and also actually in Pennsylvania. And where are the facilities where they manufacture the vaccine, that I don't know. 90+ vaccine in development. Is it common to have multiple vaccines for a single disease? Yes, and actually in this case, it is also very important. Again, because everybody needs to be vaccinated, 7 billion people, minus the children in the moment, all at once. And so it is very important because different kind of people may be allergic, like people who have autoimmune diseases, people who are allergic certain components of certain viruses, vaccines. For instance, the Pfizer and the Moderna vaccine contains polyethylene glycol, or PEG. And many people are actually allergic to PEG. They even can get an anaphylactic shock from PEG. So people who are allergic to that should not get those vaccines. They should get the Johnson vaccine or some other vaccine that doesn't have that. So it is actually very good to have a number of different kind of vaccines. Will booster shots be required in the future? Well, we don't know yet. So because we just started to study the virus and the vaccine against it, these trials really have to run out for more than 2 years in order to be able to tell that. So we have to be able to tell by then how long the protection lasts, and then we will be able to tell whether booster shots will be needed or not. It is possible, but it's also possible that it will last for a long time. Like if you think about the tetanus shot, right? You actually have to repeat that every 10 years. So we have to see how is it gonna work. We don't know the answer to that yet. Should one get the first available vaccine? Yes. So I am actually, I work for Janssen. My team actually did work on this vaccine. We did the non-clinical project management on this, and I got the Pfizer vaccine. I already got the first shot, and I'm going to get the second shot on March 25th. So whatever is available for you at the time, then you should take the one, and you will not be able to have a choice because they are all for the emergency use. As soon as these vaccines are going to be approved formally and finally, once those 2 years long trials are going to be completed, then you will have a choice which vaccine you want to get. Like for instance, if you have to get shingles, you have a choice whether you want to get the Merck vaccine or the GSK vaccine. And so once these vaccines are going to be approved, like finally and for general use, then you probably will have a choice, but you really don't have a choice in the moment. And the vaccines are distributed by the governments. So the companies really, once they release them, they release that to the care of the governments, and we really don't know how the government distributes. I mean, we don't know, but we don't have any influence on that, how the governments are distributing it. What are we like to see for children? So it is very important also, of course, to vaccinate children, but in any normal drug development, usually, unless it is a specific child disease, usually they start with adults. And then once they see that the drug is safe in adults, then they usually move to the pediatric use. In this case, of course, it is very important, especially in adolescents. So the children, especially who have underlying health conditions, can get actually the vaccines from age 16. But below that age, it really needs to be, again, studied in trials. I cannot speak for the other companies, but my company already started the pediatric trials. They actually already dosed the first adolescent children with that. And so in a few months, again, it has to run out for a few months until we will have enough data to be able to apply for the emergency use application for children as well. But it is just as important, of course. Yeah.
Dr. Antal, are there other common misconceptions that prevent people from seeking the vaccine? Misconceptions that you think are important for us to get the word out about?
Well, there are two that I can think of. One is that I hear often is that people say that if I will get sick from the vaccine, why should I take it? Well, again, you get sick from the vaccine. You might have a side effect from it for like a day. You are not going to get sick from it. You just have a side effect from it. You will have a little fever, or you will have headache or nausea. Usually, these are the most common. There is also a common side effect of the Moderna vaccine. If you are going to get that, first of all, the Moderna vaccine has 3 times the concentration as the Pfizer vaccine. So it's a little bit stronger, and that's why the side effects are also a little bit stronger. And one of the strong side effects in women is that they actually, because we get it into our arm, it actually is going to, because of the immune response, some of the lymph nodes under the armpit actually going to enlarge, which will take a while until they resolve itself. And if somebody happens to also due for a mammogram at the time when they got the vaccine, they oftentimes can actually mistaken that, that something is wrong. And so there are some scientific papers on this. I think the medical community is aware of this. And so I am just saying that people should not be alarmed about that. That's, again, something which is a natural response in your body when you are working with the virus. With the vaccine, and it is going to be, it resolves itself eventually. The other thought I have is the natural fear of community of Black and brown population, that they are afraid of getting any sort of new medication, because of the history of, unfortunately, those population being used for medical experiments in the past. I have to assure everybody that especially Johnson & Johnson is taking a very, very large care to make sure that the composition of the clinical trial participants is actually extremely diverse. We have a special program for diversity and inclusion in our clinical trials. So we make sure that the clinical trials are reflecting on the population, ethnicity, distribution of the population where the drug is going to be intended to use. And the data is absolutely the same, regardless of gender or ethnicity or race or anything like that, and people should not be afraid to get this vaccine anywhere.
Are there other questions for Dr. Antal?
Well, thank you very much for listening. I hope it was useful, and stay safe. Take care of yourself and your loved ones. And I am very sorry if you have been affected by this terrible virus, or your family was affected, or you had a loss. I am really, really very sorry for that. But I am really hoping that everybody is going to consider taking the vaccine, and we will get through it, and we won't have to at least suffer from this any longer.
Thank you so much. And thank you for your time this morning for this informative presentation. It's nice to hear from someone who has had a hand in developing these medications and helping them to be launched and available to us. I'd like to close us with a prayer. I'm going to share my screen just briefly. Let us pray. God, you are with us all the time. All the time you are with us. Today we remember how things used to be. We remember how many things we have gone through. We remember things we missed and people we lost. Today we hope. We hope for healing. We hope for vaccines. We hope for wisdom. Today we share. We share smiles with one another. We share our joys and our sorrows. We share our dreams for the future. God, you are with us all the time. All the time you are with us. Be with us as we remember, hope, and share. Amen.
Amen.
And we'll pray for your family. Thank you for sharing their names, and they'll be in our hearts, and we'll pray for them. And we'll pray for you and your colleagues as you continue your efforts. So thank you again, Doctor. Thank you so much.
Thank you so much. God bless.
Thank you.
God bless you, Ildiko.
Bye-bye.
See you soon. See you soon.
Bye-bye.
Bye.
And thank you, Deacon Bridget, for facilitating the special presentation today. And thank you, friends, our studio audience here on Zoom, others on Facebook who may be watching this or a future recording of this. And let's all do our part to reach out our hands in love to our neighbors, helping them to get access to the vaccine by registering, by signing up, by a ride, whatever we can do to help people have access to healthcare, and also not to be alone in this time. And how wonderful that would be just for someone to hear from you today, a phone call. Even if there's no practical way you can help them, they would love to hear your voice and feel less alone. And that would be a source of hope and healing for them, I'm sure. So God bless you all, friends. Take care. And we'll see some of you this afternoon, 4 o'clock, for our church scavenger hunt. It'll be fun for kids of all ages, including me. See you soon.
Bye.
Bye.
Thank you, Dr. Antal.
Bye-bye.